Welcome, everybody. Glad you guys decided to stay toward the end of the lunch session here for my talk. Diane Retallick. I'm president and COO of Primrose Bio, and happy to tell you guys a little bit about our Pfast protein expression platform for accelerating antibody discovery. Okay, so a little bit about Primrose. We're located here in San Diego. We were formed in 2023, but the Pfenex platform has been around for a very long time. We have about 25 years of history with the platform, expressing a wide variety of different proteins. And we focus on end-to-end strain engineering all the way through process and analytical development for GMP manufacturing. What I'd like to start off with is telling you a little bit about our platform. It was purpose-built for protein expression. So unlike other systems that were lab cell lines that were adapted for protein expression, we chose this platform based on Pseudomonas fluorescens expressly for making proteins, and built a very comprehensive toolbox around that and coupled it with a high cell density process that is scalable from half mil scale all the way through 100,000 liter scale. Now, a little bit about the platform. As I mentioned, it's based on Pseudomonas fluorescens. This is a gram-negative bacterium. What we've built is a very flexible toolbox. So we have a very large collection of off-the-shelf expression plasmids that we use for tuning expression of the different proteins. To compatible plasmids, everything's antibiotic-free, animal origin-free. So these are maintained by complementation. And then we have sets of tools for tuning transcription and translation, as well as a really large collection of secretion signal peptides that really help us to not only move the protein to the periplasmic space where disulfide bonds can form, but also these can have a really large impact on our titer and the quality of protein expressed. We can also impact quality with different host strain modifications. So in addition to our off-the-shelf plasmids, we have an array of different host strains, all derived from a single progenitor strain that either overexpress different folding helper proteins, chaperones, disulfide isomerases, and things like that, or maybe carry one or more deletions of different proteases that can negatively impact the quality and titer of your protein. More recently, we've been focused on adding additional features to this toolbox, including non-canonical amino acid incorporation for site-specific conjugation, more tools for further improving folding, and also tools for streamlined recovery. Now, just to give you an idea about the impact of some of these tools, here is an example where we're expressing, this is a monomeric target protein, screening a number of different secretion leaders. So here about 39 different secretion leaders. Looked at 2 different host strains, and you can really quickly see from this CGE image how the different secretion leaders can impact the expression from nothing to really high levels of expression, as well as the quality of the protein. Here in this example, number 7, showing us very high titers in our half mil cultures of about 2 grams per liter. Now, how this workflow is operated, we do a pooled cloning for our Pfast workflow. So we'll take a single gene encoding the protein of interest, ligate that or combine that with our vector pool. This could be up to 200 different expression plasmids, and then typically transform that into just 1 or 2 of our different host strains to get an idea up front of which of the tools, which of the expression tools are really helping us to drive high titer, high quality. We use NGS to confirm the plasmid representation in the pool and just as a QC check. After the transformation, which is all done in high throughput in 96-well format using electroporation, we then plate onto selective media, pick those single colonies, and then pick those into our growth plates. Then after those grow up in a half-mil culture, we then seed them into our growth and production media, typically grow for a day, induce for a day, and then we can harvest those, lyse, and then rank expression by using either high-throughput SCS-CGE or ELISA. And then we can ID our hits. From there, perform enrichment, so we can get a better idea of the quality of the protein, provide material for more in-depth analysis, and then sequence out any hits from there to move forward into scale-up. So really quickly, in about 10 days, we can move from clones to selecting optimized hits out of that screen, or identify subsets of tools that work really well for that particular modality. So in the next couple of case studies, what I want to show you is 2 different ways to use this workflow. In this first case study, we're focused on identifying the tools and scaffold that are going to be used for optimizing Fab expression. And this is really used in combination with our partner's computational system for optimizing antibody binding. So this was operated in 2 steps. First step really focused on identifying the toolbox components as well as the scaffold that the lab wanted to use to then build their computational designs. We took 3 Fab scaffolds, ran that through our Pfast screen, identified a tool set that was then combined with 48 different computational designs to evaluate how those new designs would impact the Fab activity. The way we approached this was first looking at 40 different expression vectors in combination with operons encoding those 3 different Fab scaffolds. It was very important for us to also look at the operon structure, so whether heavy chain or light chain came first. So in this first screen, we looked at about 240 different strategies to then, in 10 days, identify what's going to give us the highest expression level to then screen our library. Sample analysis worked pretty much the same in both steps, where we would grow these in our high-throughput expression media, prepare our samples, and then evaluate first step using SDS-CGE, and then after we identified the scaffold that worked well, evaluating those 48 different clones, first looking at titer, and then also activity. So now going to that first step, here we wanted to identify which scaffold and which expression strategy was going to work best for our library screen. And it was pretty clear from the start which one was our winner. So Fab version 1 scaffold was a clear winner over the other 2, and in particular using the heavy chain first, then light chain operon design. So here in that initial screen, we identified expression strategies that gave us Fab up to about a ½ gram per liter in that 96-well half-mil culture. We took the top 24 to then sequence using NGS to identify which particular expression strategy, so which secretion leaders were giving us the highest levels of expression. So here, diving into those results a little bit further, we see that there were really 2 secretion leaders tied to the N-terminus of the heavy chain that were giving us the highest levels of expression, leader 3 and leader 11. And those were the 2 that we then selected for screening our library further. So now, combining those selected tools, we looked at 48 different variants here in the 2 expression vectors and evaluated 3 different host strains. So now bringing in some additional tools for secretion as well as disulfide bond formation to see if those would further improve our titers. Now, in this screen, we're evaluating not only how much Fab are we making, but also are any of these variants giving us higher levels of activity? So first, looking at expression, we can see that in host strains 1 and 2, we were able to further boost expression of some of the Fab variants up to about 1 gram per liter in that 96-well plate. And when we further evaluated by ELISA, we could see several candidates that showed us several-fold improvement over baseline activity. So in about 8 days, we were able to screen through these 48 variants and identify several lead candidates for further development. So hopefully able to show you here how, involving some additional analytics early on to look at quality, activity, gives us very quick and good evaluation at high throughput. Here we can quickly identify those leads and get ready to scale those up for production. Now in this next example, we were evaluating a Fab VHH, and in this particular example, really focused on identifying an optimal production strain. So again, here we want to screen our toolbox to identify those strategies that are giving us high-level expression. In this Fab VHH bispecific, we had the added feature that there's no disulfide bond in between the heavy and light chain. So as we looked at the 2 different operon strategies, we first grew those up, expressed them, evaluated those by SDS-CGE. Here we looked at it just under reduced conditions since there was no disulfide in between the 2 chains. Then evaluated ELISA to look at activity and intact mass. So here in the SDS-CGE, just want to give you an idea about how we are calculating the titers. We're looking at heavy and light chain expression, evaluating that using our CGE reference ladder. Once those 2 are quantified, then we want to look at those to look at the ratio of heavy and light chain together. We want to see nice even ratio of titer. And you can see here the importance of screening those different operon designs from the start, because whereas in our first Fab example, it was important to have heavy chain at the 5' end of our operon, here it was very important to have the light chain. You can see we can have much higher titer with light chain first in our operon, giving us about a little over 0.5 gram per liter in that half-mil culture. Now, when we looked at ELISA, here we're evaluating using an indirect ELISA with a single dilution factor. Unfortunately, we were making so much Fab that we kind of pegged out the top end of our curve, but it validated for us that of those highlighted in open triangles or green, or open diamonds or green diamonds, which showed us high titer, we were getting also really nice ELISA activity. When we enriched some of that material, looked at intact mass, we can see that in the 2 example samples from strain 1 and strain 2, we have fully assembled Fab VHH with the appropriate intact mass of about 62 kD, and a little bit of unpaired chains as well. But overall, really nice quality and high titer for initial screening and expression to identify our different screening strategies. All of that could be accomplished in about 10 days and really allows us then to clear a path to be able to scale up, take that into higher cell density fermentation, and produce enriched material in about 5 weeks. So now I'm going to switch gears a little bit to some of the things that we're working on to further streamline these workflows. And one thing, of course, would be easier recovery of the protein. So there are a couple things important for expressing antibody derivatives in microbes. It's really important for it to move into the periplasmic space so that we can form disulfide bonds. So One of the things that we're evaluating in moving proteins all the way out for easy recovery into the extracellular media is a periplasm first pathway. So we get those proper disulfides formed, and then an extracellular secretion mechanism moves it into that cell-free broth, moving it away from intracellular proteases and other things that may impact product quality. So obviously getting it outside of the cell allows us easier recovery, lower host cell protein, host cell DNA contamination, as well as lower endotoxin, and can offer us the ability for real-time expression monitoring by just removing some of that cell-free broth and avoiding that lysis step. So what we've been able to do is identify several different host cell targets that allow us to move proteins of a wide variety of sizes out into the cell-free broth. So what we're looking at here in these expression results, so we've got SDS-CGE analysis of different proteins ranging from about 15 kilodaltons to 60 kilodaltons, where we're either looking at the cell-free broth, so cells spun down and just the medium, versus the pellet, then resuspended, lysed, and the soluble fraction. The EXT123 are 3 different types of modifications we've made in our host strains, and you can see in the wild-type controls, no protein or very little protein moved out into the extracellular space, but when we make these different modifications, we're able to move a significant amount of the protein that we expressed out into the extracellular broth. And this will really allow us to then further streamline our screening process. Of course, very important is the quality of the protein that we get out. So when we look at each of these by intact mass, we can see that each gives us the expected mass for that particular protein with no modifications, except in the case of the antibody derivatives where we get some pyroglutamate formation since the end term was glutamine. All right, so before I wrap up, I just want to tell you a little bit about P. fluorescens in general. It's a very safe organism. It's biosafety level 1. There are 6 approved products using this platform. Over the years, we've developed a really robust high titer process for large-scale fermentation and have used this for a number of different proteins to really accelerate development from hit to lead discovery all the way through manufacturing, clinical, and approval. We've worked with a wide variety of proteins, lots of different antibody derivatives, different non-antibody binding scaffolds as well. And what we've seen in many cases is up to a 10 to 20-fold improvement over other systems like E. coli, CHO, or yeast. And then just finally, a little bit about our track record. As I mentioned, there are 6 approved products that are made using the Pfenex platform. We've done a wide variety of different tech transfers to different CDMOs around the world, as well as internally to our pharma partners, worked with a large number of different companies from startups all the way through large pharma, and really have been able to show the power of the Pfenex platform in helping to get these products onto the market. So with that, I'd like to conclude. If you've got questions, really happy to take those now. Invite you to stop by our booth to learn more. And I'll put a plug in for Chris Warner's poster where he has an example of VHH ADC produced using the Pfenex system. So if you want to learn more about non-canonical incorporation and conjugation with our different tools, please stop by and talk to Chris. All right, thanks.
Hi.
He's got the mic.
If you want, I can do loud enough. It's going to pick up. So. Awesome, great.
So, quick question. Do you offer this as a service, or would you also set it up at a customer's facility?
We do offer it as a service, and as for setting it up in a customer's facility, that's definitely something we're open to talking about.
Awesome.
Yes.
Thank you very much.
Do you have any cell-free protein synthesis from the Pfenex?
Yeah, that's a great question. That's something that we've thought about developing, but just haven't had the bandwidth to do. But I think it would be very interesting, as you know, Pseudomonas is a really robust organism and tends to have a lot of features that other systems don't. So I think it would make a really nice cell-free system as well. Yeah.
Thanks.
So this primrose is BD guy.
Go ahead, Lou. Lou's been pushing me to do this for, I don't know, 5 years. Right. Any other questions? Okay, thanks.